Many people around the world rely on sheep, goats, and cattle for food, milk, and their livelihoods. These animals are susceptible to many diseases, including one studied in our lab called peste petite ruminants virus, or PPRV. This highly infectious virus causes illness and death in affected herds. PPRV is currently in Africa, the Middle East, and Asia, and can impact 80% of the world's sheep and goats. To determine if PPRV is present in a certain region, scientists look for evidence of disease by collecting blood samples from sheep and goats. The serum portion of the blood is then tested for antibodies to the virus, which indicate that an animal has been exposed to the virus. This lab test is called a competitive ELISA. A 96-well plate is first coated with a solution of PPRV antigen, which makes the walls of each well appear similar to the outside of a real PPR virus. If PPR antibodies are present in the sheep and goat test samples, they will bind to this. The coated plate is loaded with test and control samples. Control samples have a known response. The positive control sample contains PPRV antibodies and the negative control lacks PPRV antibodies. Additionally, a solution of the control antibody is also added to compete with the natural antibodies in the test serum to bind to the wells, hence the name competitive ELISA. Once loaded, the plates undergo 2 incubation steps and washes, the addition of a second solution that binds only to control antibodies and allows for color development, and a final 15-minute color development step. If there were PPRV antibodies in the test sample, the well will not change color. This means that the natural PPRV antibodies will have outcompeted the control antibody, bound tightly to the walls of the well, and not bound to the second solution. If there were few or no PPRV antibodies in a test sample, the opposite will occur and the well will change color. A plate reader machine is used to determine the level of color in each well and determine if a serum sample is considered negative or positive. Once results are compiled, scientists have a picture of the population exposure to the PPR virus in a given region. These results may show geographic variation in exposure, or that other factors may influence disease patterns such as species, age, and gender. This knowledge helps veterinarians and scientists better target disease control, prevention, and vaccination strategies.